Hello, and welcome to the first session of the Arcadia GoodHemp Industry Information Series. Today's talk, Cannabis Breeding, Past, Present, and Future, and Why It Matters to Hemp Growers. Thanks for taking time out today and being with us. My name's Sarah Ryder. I'm the Chief Commercial Officer at Arcadia Biosciences, and we're really excited to have you join us. The Industry Innovation Series was spawned out of the idea that hemp and the related crop industry is evolving at a much faster rate than any other crop, and therefore there's a need to provide key information in a really fast manner consistently to help growers and the rest of us in the industry gain credible information to help drive our decisions. Our series is intended to deliver value on a regular basis through topics that are important to you. Please continue to join us in future sessions. Today's session, Cannabis Breeding, Past, Present, and Future, Why It Matters to Hemp Growers, is going to be presented by Arcadia's Chief Technology Officer, Dr. Randy Schultz. Dr. Schultz has been instrumental in accelerating our novel non-GM hemp varieties with Architect, our proprietary rapid prototyping and development platform. He brings many years of experience in genetics in other crops like corn and soybeans to hemp. As CTO, Randy oversees the execution of our strategic multi-year plan to become a market leader in high-value hemp innovations, as well as continuing to advance next-generation products through our company's Good Wheat pipeline. A final point of housekeeping. We welcome your questions, and we'll plan to address as many as possible at the end of the session today. At any point during the presentation, please place questions in the Q&A module by clicking on the Q&A icon as shown on the screen. Again, thanks for joining. We look forward to sharing with you over the coming months. Continue to visit our hempinnovationseries.com for updates and additions to the series. I turn it over to you now, Randy.
Thank you, Sarah. And good morning, good afternoon. Thank you all for joining. I know this is a busy time with probably a lot of you, prepping for the spring planting season. So we really appreciate you taking some time. This is our kind of a first seminar in the series. Get my video on here. The first seminar in the series. So we're still getting to know our audience, and hopefully there's something for everyone, whether you're new to growing hemp, thinking about growing hemp, or have some experience in growing hemp. And we're here to answer questions both today, and if there's anything that we don't address or that you're looking for more information from, please feel free to reach out. We're happy to help and engage and help people be successful. So with that, I'll start with just a little introduction to Arcadia. So Arcadia has been around since 2002. We're headquartered in Davis, California, but we do have locations across the U.S., particularly in Phoenix. We have a research station in Idaho. We have a operation in Hawaii, and then we have employees kind of spread across the United States. So we're focused on kind of two major objectives. One is developing and bringing the best genetics to farmers to help everybody be successful. Sarah mentioned our Architect technology platform, which I'll talk a little bit about a little bit later in the talk. And we also think about bringing value to consumers through healthy ingredients. And a good example of that is our wheat portfolio, Good Wheat, where we have attributes such as extremely high fiber, reduced gluten and allergenicity, as well as increased shelf life, with the objective of really bringing nutrition, wellness, and high-value specialty products directly to the consumer. So we do work in a number of different crops. Hemp is fairly new to us. We started working in hemp a little over a year ago. So we're still learning along with the rest of the industry. And hemp will be the focus of my talk today. So when we got into it was actually our wheat growers that kind of brought us into the hemp industry. A lot of large-scale wheat growers were interested in using hemp as part of their rotation. And as they got into experiences in growing hemp, they experienced a lot of challenges and engaged with us to kind of work through those, which kind of dragged us into the hemp industry. So this is a pretty much a very new crop. And as Sarah mentioned, things are evolving really rapidly, and there's clearly a lot to learn, which really, makes it not too surprising that there's a lot of challenges and pain points. So quick kind of backdrop on some of the key legal milestones that have made this crop federally legal now after about 50 years of being illegal. So in 2014, the Farm Bill legalized hemp. A lot of states picked up a pilot program to start to understand the crop. And then in 2018, the Farm Bill fully legalized hemp and directed the development of a national program. The USDA came out with that program, the interim final rules, it's called, for the domestic hemp program in 2019. And those, the states are currently writing their own plans or just following in line with the USDA plan, which really kicks in at the end of October. So right now in the industry, it's a little bit of a mix. There's still some pilot programs in effect. There's some states with a little bit of unique attributes to their program, and then there's other states that'll just fall in line with the USDA guidelines. The other major thing is kind of lack of FDA action is holding back the full potential of the industry. Now there's a tremendous interest in hemp among growers. In 2019, about 500,000 acres were licensed for hemp production, and we think, and the insights we have from both the wellness and grain and fiber side is that there's a tremendous greater potential than what's currently there. We see a huge opportunity as some of this regulatory uncertainty gets figured out. So just to highlight a few of the challenges, because it is so new, there's a lack of agronomic information, there's some misinformation, and a need for education among growers and really all along the supply chain. I mentioned the shift in regulatory environment. Clearly, there's a lack of good genetics. So, except for the clandestine, growers growing marijuana and breeding marijuana illegally, there's been very little breeding of this crop in over 50 years. There's also a relatively immature supply chain, which is, not surprising given the nascent nature of the industry. A lot of farmers are experiencing poor quality of seeds and clones. There's been lawsuits. We've heard a lot of horror stories. One of the main issues as farmers are producing is the risk of non-compliant crop. So hemp has to be less than 0.3% THC, or else it requires destruction and essentially total loss. There's a couple of different reasons for that. One is, the genetics haven't been optimized quite yet for that, and that's something, of course, we're working on. And also and I'll get into this a little bit later, there's off types in a lot of the commercial seed that's sold, which contributes to that risk. Weeds, pest, pollen control. This crop has really not been bred, at least not the high CBD varieties, for large scale mechanized farming, and so you see a lot of people what we call gardening. It's a high labor, hand harvest, hand weeding, and the other thing is uniformity and regional adaptation. So because it's so new, there hasn't been the time for people to really breed regionally, and that's something that I'll touch on a little bit later as well. So a little bit about this plant. First, I'll start off that cannabis and marijuana are from the same species, Cannabis sativa, and hemp is legally defined as cannabis with less than 0.3% THC. So really, all hemp is cannabis, and it really only becomes illegal in marijuana when the THC content rises above 0.3. So it's critical that growers in this industry are able to maintain that threshold to be successful in this industry. So cannabis is a really interesting species. It's somewhat unique among other large-scale crops. It is annual, fast-growing. Most of the varieties are induced by short day to flower. There are some autoflower varieties, but the majority of them are photoperiod sensitive. The seeds are really high quality protein and oil, which is a huge market potential going forward. And the biomass has a tremendous number of industrial uses. It's fairly low maintenance to produce, although I think people are learning that it may take a little bit more TLC than was originally thought. Interestingly, it's dioecious, so separate male and female plants, and just like us, the males have XY chromosomes and the females have XX chromosomes, which does pose some challenges for breeding. It's also an obligate outcrosser, and the pollen's really light, travels quite far, so there's isolation issues, and farmers need to really watch out for inadvertent pollination and being good stewards and good neighbors. The plant is well known for being tremendously productive in secondary metabolites, the cannabinoids, terpenes, other molecules that have a lot of use in wellness, but also poses some challenges in maintaining compliance. Stress can induce hermaphrodites, which has been an issue in some cases, and can result in reduced yield if a crop is inadvertently pollinated. So we think of it as semi-domesticated. There's certainly a tremendous opportunity to continue to breed and adapt this crop to provide pretty significant increases in yield and other attributes. So cannabis has been with humans for a long time. As a matter of fact, it's thought to have been domesticated about 12,000 years ago. And speaking to the utility of this crop for a variety of reasons, it really, humans carried it through all of the migration routes. Really globally, it can be found both feral and grown for industrial or medicinal uses. There's kind of two main types. So hemp, as we think of it, is really what we call industrial hemp. It has been bred for low cannabinoid levels overall. There's kind of two groups. You have the European hemp and the Chinese hemp. So all of the fiber and grain varieties would fall into this hemp hybrids group. And then you have kind of the marijuana hybrids. These are the high cannabinoid levels that have been bred specifically for medicinal uses primarily. And the current high CBD, high CBG varieties in the hemp wellness market were developed by crosses between hemp, these two classes, the hemp and marijuana hybrids. Interestingly, some genomic analysis shows that these varieties, these high CBD varieties, are actually mostly marijuana type genetics. So if you look at this figure on the left-hand side, you'll see three different colors. So the yellow cluster, so this was developed by looking at a whole bunch of genetic markers and clustering things together that are similar. So the yellow are industrial hemp varieties, the blue are marijuana drug type varieties, and the red are feral varieties or naturalized varieties. And not sure if it's clear to everybody on the screen, but two well-known high CBD medicinal varieties, hemp varieties, CBD RX and Skunk One, both cluster really clearly with marijuana types. And if you look at the panel below, the blue is the marijuana type DNA. And you can see that, these varieties, the Skunk One and the CBD RX, are clearly predominantly marijuana type genetics. And what this tells me is that there's a huge opportunity, because marijuana was really bred, in clandestine, underground, for indoor grows or limited outdoor grows. It really hasn't been regionally adapted, and there's a huge opportunity to use modern breeding tools to rapidly improve this crop for different geographies and different attributes. So if we take a closer look, we can see that, so on the left-hand side here is the cannabinoid synthesis pathway. You don't need to look at all the details here, but it's important to note that the CBGA molecule is the precursor to both THC and CBD. And those are driven by what you call synthase genes, CBDA synthase, THCA synthase. So chemotypes are really just chemical phenotypes, and there's really 4 different chemotypes in hemp and cannabis. So type 1, you have much more THC than you do to CBD. So this figure in the middle here on the right-hand side shows you where these synthase genes are. They're actually all closely linked on a single chromosome, actually in a fairly low recombining region. So the type 1s are high THC due to an inactive CBD synthase. Type 2s are about equal in THC and CBD because both of those genes are functional. Type 3s, you have a much higher CBD than you do THC, and this is due to an inactive THCA synthase gene. And then type 4s, which are on the market this year, they're relatively new. Those are high in CBG because you have a non-functional of both the CBD and the THC. So to be a hemp grower, you really have to grow either type 3s or type 4s, because if you get the other types mixed in, you're likely to be non-compliant. And as a grower watch out, we've been analyzing a lot of different seeds from a lot of different providers, working with our own material. And, as a watch out, we see many, what seem like reputable seed providers delivering really some significant segregating off types. So if you look to the right of this graph, which is just showing the different types based on chromatography analysis, you can see it ranges from, we've seen as high as 32% in seed down to 0, and everywhere in between. So, definitely keep an eye out, ask those questions, make sure that when you're buying seed, you're thinking about data supporting that there's minimal off types. So what we do is we have in-house chromatography, and this is very useful for both our breeding program and our seed production program. For example, we're producing pre-commercial developmental variety seed this year here in the Davis area. Every single parent involved in those crosses and that seed production, we've done chromatography analysis on and ensured that any off types were removed from the population, and that enables us to deliver really high quality, all type 3 or type 4 seeds in the next generation. We also use it in our breeding program. We're able to drive, better ratios, higher cannabinoid levels, lower THC levels by monitoring, plants through the breeding program. And it's also useful for helping farmers determine when to harvest their material and still stay compliant. So we do weekly monitoring, and we're working with a number of farmers on this for this summer to make sure that we can predict when the optimal harvest time is. So another watch out for growers, we've also found that uniformity is really an issue in some currently available varieties. So on the left-hand side, this was commercial seed purchased. On the left-hand side, you can see you get anywhere from small bush to large bush to runts. And on the right-hand panel, you can see what a nice uniform variety looks like, where everything's really flowering at the same time, the same height, much more uniform field. And then the center panel, this is just another example at the seedling level. This is what we're working on. You can see some commercial varieties showing a lot of variation, germination timing, architecture, whereas one of our early varieties is showing a very uniform emergence and pattern. Finally, last grower watch out. Because these crops really haven't been regionally adapted or bred for different regions, we have single varieties kind of being sold all across the United States, and that can lead to some really significant disease problems. So here's an example from the Portland area this year in one of our cooperators' fields. This was a #1 variety sold in the United States over the last several years, and you can see just completely destroyed by botrytis, which is also called bud rot, whereas other lines in the field actually didn't get infected at all. So clearly there's some differences in disease resistance, and that's one of our objectives is to regionally test these, start to adapt to make sure we have the proper disease controls for the different geographies. So here's a snapshot of how we're approaching this year. We're setting up multi-state regional testing. The panel on the left shows the regions where we're currently setting up regional trials, either through our own research stations or through collaborators, trying to get a good mix of different hemp growing regions. And then the bottom right is just a snapshot from a field we did this winter down in Imperial Valley, California. You can see we've got a number of plots. This is a pretty difficult environment for a lot of these varieties, and we saw certainly a lot of varieties do very, very poorly, but a few that seem to do pretty well down there. So by taking this sort of approach, we can start to really understand where to place different varieties in different regions and different environments. So to kind of bring it together, I've been talking a lot about the cannabinoid market and the wellness market, but hemp's much bigger than that. It's one species, but there's actually kind of three different market classes. You have the wellness. You also have plant protein. I mentioned that the grain is, a really high quality protein, good oil, and really has the potential to, help drive the, the sort of plant protein movement, as well as, potentially be a replacement or a additive for replacement for other protein sources like soybean. And then of course, fiber and industrial use. There's hundreds, if not thousands, of uses for this crop from an industrial perspective, and those supply chains are continuing to evolve. So we're actually breeding for all three of these market classes, though we've started in the more in the wellness space in the short term, as we're just getting started. So now I want to transition and talk a little bit about our technology platform and how it helps us unlock some of the bottlenecks in what would be a traditional hemp breeding program. So if you look on the right, these are just some schematics. If you think about all the different variation that you see in the crop today, and kind of driving that to a highly harvestable, mechanized structure for both the cannabinoid crops and the hemp crops. To run that sort of breeding program, you need to have access to really good diverse germplasm, which is somewhat of a challenge. The USDA actually destroyed all of the cannabis in the gene banks and the germplasm banks. They are trying to build that back up. I think Cornell is going to be the site of a new germplasm database, but certainly there's some challenges in getting access to that at this point. Phenotyping and regional trialing. So in order to start to understand how genetics play a role in different phenotypes, we need to measure a lot of things in the field. And so we're standing up drone-based phenotyping and other things in order to really accelerate that process. Genomics enables us to really be much faster about making selections. We can use genomic selection and other tools so that we don't have to just plant a whole bunch of things and pick one in the field. We can make computer predictions and be much better at moving faster. And then introducing valuable traits. So part of our platform is using tilling and other genome editing and other techniques in order to accelerate the creating diversity in some of the key genes and key pathways. So this is just a snapshot of that technology platform. If you look on the right, it's really a design, build, test, learn, combine cycle. So in the design, we use genomics, bioinformatics, data scientists in order to select the genes that have the most impact on, whether it's yield or harvest index or cannabinoid profile. We then use CRISPR and tilling technology in order to create diversity in those gene targets and create a prototype. We measure those really quickly, both indoor and controlled environment agriculture, as well as outdoor, in order to understand if we've got, the optimal allele to give the desired outcome. And then the breeding program can quickly combine and stack those traits in order to basically custom design varieties. If you look on the right, things like, completely de-risking, THC, compliance issues, disease. We see a lot of opportunity in yield because these crops are not regionally adapted and are really semi-domesticated. Pollen flow is a really big issue. So there is feral hemp out there, and so even if your neighbor is the best neighbor in the world and planting only feminized material, certainly there can be inadvertent pollen flow, and that can reduce yields for the wellness market. And so we're working on some pretty interesting concepts around making sure that plants are not receptive to inadvertent pollen. So really the sky's the limit, and we're really looking forward to and are currently working with partners that have some really interesting ideas about some custom design varieties. So with that, I'd like to wrap up and open the floor to questions. And I hope everybody found at least something useful out of this time, and really appreciate you guys taking the time to join us. So I think Sarah is going to help moderate questions, and just a reminder, if you do have a question, I think you can click an icon and type that question in, and we'll do our best to answer it for you. Thank you.
Thanks, Randy. Yeah, we have a couple questions so far. You mentioned chemotyping. Can you explain if you mean you test every single plant and how that works?
Yeah, so it depends what we're trying to do, but I gave the example of our developmental variety seed production that we're doing this year. This is indoor seed production, so we're able to control pollen, almost completely, and avoid any stray pollen from outdoors or from other parts of our program to be involved. And in that instance, yes, we sampled every single plant involved, both the pollen donors and the pollen receivers, and we run them through. We use gas chromatography, which enables us to see the cannabinoid levels of both CBD and THC, as well as other molecules. And we can use that to make sure that we remove any inadvertent off types that happen to be in that population. And that's something we'll continue to do going forward.
Thanks, Randy. Another question is it important to use feminized seed?
Okay. Yeah, so it is. There's a couple of ways to do this. So I mentioned kind of early on that, cannabis has both males and females. The males produce the pollen, the females produce the flowers. So in like an industrial hemp variety, a lot of them you would plant both. If you're trying to make grain, for example, you'd have a mixture of males and females. You'd plant those. The males produce a tremendous amount of pollen, which is great for grain production. However, in the wellness market, seeds are actually undesirable. They don't produce any cannabinoids. So for the wellness market, what you really want is a seedless flower. And any inadvertent pollen actually decreases the value of that crop. So to be a good neighbor, one solution is feminized seed. Another solution would be planting clones from a female. That would be another solution. And the third one is isolation distance. So, if you can find a spot, or if counties, some of the counties like the county we're in, Yolo County, I work with them to kind of think about how different growers can coexist, because there's cannabis, marijuana type growers here in the county that grow outdoors. And so we're thinking about isolation distances and putting in some measures like that to help ensure. But yes, planting feminized seed is definitely ideal. Another option would be going with clones that are completely female as well.
Oh, I'm sorry. I'm talking into my mute button. So thanks, Randy. Another question. What is an autoflower?
Great. Yeah, good question. So most of the varieties are photoperiod sensitive, which, a lot of growers are used to. Soybean is photoperiod sensitive. When the day length starts to get shorter, in August, September, for example, hemp begins to flower. It really won't flower until the day lengths get shorter. When the day lengths are short, it'll start to flower right away. So if you take a photoperiod sensitive hemp variety, which most of them are, and you plant them in, say, an off season in Hawaii or down in southern Florida, or somewhere where the day lengths are sort of less than, say, 14 and a half hours, it's going to start to flower right away, and it'll be a tiny little plant, won't produce much yield per plant. And so autoflowers were developed by a cross with a sort of subspecies of cannabis called rudialis. Brought that trait in And it's from like really the high northern latitudes. So it sort of evolved to not be photoperiod sensitive. So what that crop does is it basically flowers on a clock. So as soon as you plant it, it grows. It grows for X number of days or until X amount of biomass has been produced, and then it starts to flower. So they're really useful in some growers' fields at different times, maybe as a second crop, for example. But they do tend to be much smaller. They don't produce as good a yield as a full season a photoperiod sensitive variety. And so, people are working on these. They definitely have a lot of utility, but you got to really think through which is the right one for you, depending on where you are, what time of year you're planting, for example.
Great, thanks very much. Couple more things. So here's a comment. It says transplanting autos has been unsuccessful. Do you have the same experience?
We do. We did that this year in Hawaii, and certainly they got transplant shock, started to flower way before they should have. I think that's fairly common in the industry. It seems like the stress of transplanting, sort of gives them that signal that they're looking for to start flowering. It could be that that's something that's just linked to that genetic region that was brought in from Rugalis, or it could be something else. But definitely interesting from our experience and from what we hear in the industry. If you're going to grow autoflower, it's best to direct seed it at a fairly high density.
And another question about autoflower. I'm sorry, the opposite actually. These photoperiod sensitive plants, if they're planted in winter, can they grow in South Florida and produce enough to grow? In other words, are there photoplant varieties that will grow in vegetative for some time, even though there are only 10 or 9 hours of light in that day?
Yeah. So this is a really great question. And part of it is the newness of this industry, particularly for the high CBD, low THC varieties that really came out of a limited number of crosses for the medicinal marijuana industry that, is really, not that old. So all of these varieties and people are working on, bringing in additional genetics through breeding programs, but, most of the varieties that are out there that people have access to really come out of very few parents. And so the adaptation to different latitudes is fairly minimal. If you look at marijuana, there's marijuana types that grow all across from the equator, all the way up into really high latitudes. So the genetics are there to be able to grow well and to produce well at a variety of different latitudes. But I think the real challenge is that true breeding programs that scale, that do multi-location testing, regional adaptation, haven't really had the time to develop those in this industry. So, one solution that some people are doing, and we're doing this in Hawaii, is to put up some supplemental lighting in the field. It doesn't take a lot. Even some fairly low power, solar powered LEDs can suffice, but it enables you to prevent the photoperiod sensitive varieties. You kind of trick them into thinking there's more daylight there. They'll put on a lot more veg, and then when you take that away, they'll go ahead and go into flowering. And you can get much better yields doing that.
Okay, a couple more questions. Thanks, Randy. Will your plants grow in Louisiana with 100% humidity and 100-degree plus temperatures?
That's also a great question. So that's something that we're very interested in answering. The varieties that we have today, we're still pretty new to this, like a lot of you are. The varieties that we have today, we have the new reliables that have been tested years in multiple environments. But really, there's very few varieties that have been, really tested in a whole bunch of different varieties. So we're moving into Florida this year. We'll be testing some of our material there. I think disease pressure like the botrytis is going to be a real issue in a place like Louisiana with high humidity. So we've seen some really good resistance to botrytis in some of our varieties, as well as to some other fungal based diseases. And so I don't know the answer because we haven't tested it there. And I think one rule of agriculture, at least for seed companies, is if you haven't tested something in a particular environment, it's very difficult to predict how it will perform there.
Thanks, Randy. A couple more questions. How far can pollen travel?
Well, that's another question. Anecdotally, if you kind of Google it and look through some literature and people will talk about, pollen traveling hundreds of kilometers if it gets in the right wind flow. The pollen is extremely viable. So, as part of our seed production, we will actually harvest pollen off of the pollen donors, feminized pollen, and we can store it up to a year or more. So that's pretty unusual. Corn pollen, for example, doesn't do that. It dies pretty quickly if it's after it's been shed. So hemp seems to be kind of unique in that regard, and it has really small pollen. It's like 20 microns, and so the wind does pick it up and carry it. And the females are extremely receptive, so they have a tremendous sort of opportunity to pick up straight pollen. So I think the isolation distances, so I think it can travel really far. I think in a practical world where you're balancing, what's realistic and what's tolerable as far as potential inadvertent pollen flow, with the county supervisors that I'm working with here in Yolo County, we think about other systems like sunflower seed production, where approximately a 3-mile isolation distance, is tolerable. You can work with the growers before the season to figure out and make sure you maintain that. For our outdoor seed production, to make sure we keep high quality feminized seed, we're looking at optimally trying to get greater than 20 miles of isolation in any specific distance. So to answer your question, the more the better as far as isolation distance, but I think we still have a lot to learn about what really the implications are if you're a little bit closer together.
We have time for a couple more questions. One is, what are the characteristics you consider for greenhouse versus outdoor field growth?
So if you're producing, say, smokable flower or some other flower product for the market, would you rather produce indoor or outdoor? I'm assuming. So I think, obviously indoor production is a lot higher cost. What we see is that the market is changing very rapidly, and I'm sure Sarah has much more insight into this than I do. For the smokable market, I think, indoor certainly has a place. You can produce some really high quality material, and the market will support a high price for that premium craft flower, if you will. I think for the general extraction market, it's going to be challenging for indoor growers to compete as outdoor growers really scale, learn how to manage some of the challenges, and are able to produce high cannabinoid biomass at a fairly low cost. So I hope that answered your question. If not, feel free to follow up or connect with me offline.
And the next one is, what are some of the challenges come harvest time? And are you doing anything about improving the ability to harvest this crop?
So we definitely think about harvestability. We think about harvest index. We think about planting density. The stems on these plants can get literally the size of small trees. And so you can manage that somewhat with planting density. There's genetics involved. We're certainly driving a breeding program towards enabling existing mechanized farm equipment to be able to efficiently harvest this crop. Some other challenges with harvest, I think, a lot of farmers are still sitting on biomass, so there's the immature supply chain. Certainly you have to dry and store appropriately or the product can degrade. And there's some infrastructure that really, still is fairly immature. I know in the Southeast, a lot of the tobacco farming infrastructure has been repurposed, which makes a lot of sense. So it kind of depends where people are as to how they're managing this. But I think the sort of gardening, hand harvest, probably will disappear as the really big growers figure out how to mechanize this as a crop.
And one last question before we run out of time. Have you tested your genetics here in Florida, and are you producing your genetics in lab or out in the field?
So we do both. We have not yet tested our genetics in Florida. As you know, Florida's opened up fairly recently. We are very interested. We're having lots of conversations. I think it's really likely that we're going to, make a move down in Florida and start testing genetics in collaboration with universities and collaborators, as well as, get them in some get some of our varieties in farmers' hands, hopefully this season. Excuse me. As far as breeding and development, cannabis is pretty interesting because you kind of need both. You need indoor controlled environment and you need outdoor in order to do your selections. So we do all of our crosses, our breeding in order to ensure purity and not that stray pollen flow. We do all of our crosses indoors in chambers, highly controlled environments. But unfortunately, in a greenhouse, you can't do effective agronomic selection. So, we go out in the field, we go to different environments, we perform selections outdoors in the real environment in order to find those individuals that are disease resistant, perform well in that environment. So you kind of need both. You need both indoor breeding. Of course, we take samples, run GC on almost everything. We sequence a lot of genomes. We do more. We use genomics to accelerate our breeding program. So it's kind of a mixed bag. You're indoor and you're outdoor. But indoor certainly is required in order to ensure the purity of the lines as you move through the breeding program.
Great. It sounds like there's a couple more questions on autoflowers, and maybe that should be a topic in one of our future seminars. We're doing these every couple weeks, so thank you all for joining and for the great questions. We're next scheduled to do one that's focused on feminized seed and really a lot of talk about pollen and pollen travel. So if y'all would like to join us on Thursday, the 28th of May, again at 10 Pacific or 1 Eastern, we'll be talking about feminized seed and the impacts on your neighbors and your profits from using that. And Randy, thanks very much for hosting today and for prepping everything. And then thank you everyone for joining us.